don't know how these guys know the science and everything. So it is more science. So everywhere you can see a lot more science. And finally, what happens because of time, we are not able to tell the science. We are looking at it in a different way. Construction of all those things, everything is based on science, and that too based on planetary motion and everything. But science, because that's what I usually used to tell a story. In South India, especially in Tamil Nadu, there is a temple in South, it's called as Palani. Palani is the name of the place. They used to have an idol. That idol is made up of heavy metals, different composition of 9 heavy metals. And that too, it's having too much composition of arsenic in it. It was made by a sage called as Bogat. So what happened? They do, if you look into South Indian temples, they used to do a lot more abhishekams or puja. During puja time, they will pour everything over the idol and they will distribute it as prasad. But for this idol, they will never use oil. For the rest of the idol, they use oil as one of the things. Here, they never use the oil because there is a process called as condensation. Condensation takes place. So after that, they will pour milk over that, and the milk will mix with the condensed water vapor, and then it will come out. So if you take that milk in a little amount, that is prasad. It is good for health because it is poisonous milk, and even poison can be used in a very mild amount, which can be treated as a medicine. So this was done in 2000 years before they have done that thing. But late 19th century only, 1996 or 95 only, they started bringing heavy metals for cancer treatment. Everyone knows about cisplatin. Cisplatin is one of the drugs used for cancer. If you look into our traditional medicine and everything, they used to have cure for almost everything. But in due course of time, foreign invasion and everything, we lost completely a lot more things. And especially the medicinal thing also we lost. But when you compare that with China, they are having well-documented evidence of almost all the traditional medicines. Actually, it went from India through Tibet to China. During Buddha era, so a lot more scholars, they came from China to learn Buddhism and all those things. At that time, they have taken all the books which are related to medicinal related, and it went there. So now we are not having almost this. Whatever I am telling is evidence based. These are all evidence based. Whatever I am telling. So there are a lot more stories used to be there, but we lost everything. Everything, even if you look into that construction wise, they are pretty good. Architectural construction of these types are entirely different. I don't know how they move the marbles and stones to construct everywhere in this country. Not only I'm talking about South. Even if you go to Gujarat or if you go to Puducherry is around, it started 1100 and 1200. Before that itself, even Nalanda University was one of the oldest university. Before Nalanda itself, I'm talking about before Christ. Takshashila used to be have a very good university. So everything is documented by Westerners. So we used to have a tradition of education and everything. So in due course of time, whatever comes from the West, now we are accepting everything. And now we are teaching that thing. So that's what, now the current scenario is entirely different. Whichever is coming from the West, we need to accept that thing. That's what I'm teaching, whatever you call it as pharmacogenomics and proteomics. Today we are going to look into proteomics tools. Proteomics tools, actually previously we are discussing about genomics, because we are starting with the gene sequence. From the gene sequence, what are the problems we identified, and if there is some mutation or SNP is taking place, how we are going to identify it. If we are going to identify it, what will be the cure? That part of thing we have studied. For identifying those things only, we are using microarray, and then differential display, and then serial analysis of gene expression. These are some of the things what we have studied. So you will have the question that how it is cross-checked. Yes, microarray, when they implemented, they got a lot more data. And then they went back and did northern blots to identify whether it is the right DNA or not. So they have done all the cross-check, and finally it is commercialized. So those are the tools for genomic analysis, gene-related things. So after that, once the transcription is done, now we need to look into the proteins. So what will happen if there is some malfunction is happening in a particular gene? It will have a bigger scale and a bigger problem if you are looking at the protein sequence. So if you want to find out what type of mutations are taking place in the protein, you need to identify some of the tools to find out what will happen and how we are going to identify it. So for that only we are having different tools, what you call it as for proteomics, we are having this MALDI. So mass spectrometry, where we can identify the protein sequence and which are all the protein involving in that expression. So for that, if you go one step back, we will go and run it on a 2D gel electrophoresis. As I already told you in the previous class, 2D gel electrophoresis is nothing but it is a two-dimension one. One is based on the molecular weight, another is based on the isoelectric point. Each and every protein is having a particular isoelectric point or charge. Based on the charge only it will mobilize. So if you look into a 2D gel electrophoresis, you will see a lot more bands. So you can take the bands or cut the bands out of the gel, and you can subject it to this mass spec and find out the sequence. So for this purpose, whatever I'm talking, it will be easier. So it was started long back, and from 91 onwards, the instrumentation and the other programming part became very nice, and we have started working on it. So how we are going to identify those things? So what are the softwares behind that? So first you need to take the protein out, and what will be the methodology, how we are going to do the experiment. That is the first basic thing. So if you look into that mass spec, it's mainly running on charge to mass ratio, M/Z. So actually it started in very late 66. J.J. Thomson itself started this thing, mass by ratio. And from that time onwards, they are working on it. Finally, in 1990s, only it started picking up. And now both the tools are pretty good. And you can use it for small molecules, that is organic molecules. And then you can use mass spec for protein also. So how we are going to use a protein? There are two types of things. One is called as the laser chip will be there, and another one is called as the ionization treatment. So everyone knows about that Millikan oil drop experiment. Millikan oil drop experiment is nothing but we are going to adopt the same principle here. Just if we are spraying the protein, it will get ionized and then it will travel. So for that only, we are calling that as time of flight. And then if we are digesting the protein into smaller fragments by using different types of enzymes, that is, if you are using trypsin and chymotrypsin and that a lot of enzyme mixtures, and if you are putting it in the protein will be chopped into smaller pieces. And then based on the time of flight, heavier will be falling at the first itself, whichever is lighter will have a longer time of flight, travel. So based on the thing, yeah, multi-turf, it is multi-turf only. Based on time of flight only, we are going to calculate the fragment size, and it is compared with the library. We are already having the standard libraries. Based on the libraries, you know about 20 different amino acids, and based on the digestive fragment, it will match, and then you will get the output. So if you are doing it multiple, we are doing it means the system, the instrument will do it for multiple times, and finally it will give you the result which is foolproof. Why I am telling this, I have done this thing and it is pretty good. So when I am doing my postdoc, I was working with a viral protein, and then the viral antibody was there. So there is no sequence for that antibody, but I grown that hybroma cells, I have purified the antibody, and then I digested it to get the FAB fragment. I got the crystal and I diffracted very well. So if it is diffracting very well around 2 angstrom wings, you can see the amino acids, the side chains of the amino acid clearly. So if it is phenylalanine, it will be phenylalanine. If it is serine, it is serine. It will be exactly, you can see the density, they call it the electron density. It looks similar to that of the side chain, perfectly it matches. But I need to have a proof, because they will tell there may be some ambiguity in the model, whatever I used for getting the protein structure. So I went back and I have taken, because if I have taken the hybroma cells and I did it, and I asked them to sequence it with the virologist, they sequenced it. The problem is, if you look at the antibodies, antibody is having both heavy chain and light chain. You know about FAB fragments. I hope you know that thing. Antibodies used to have heavy chain and light chain. In heavy chain, it used to have 3 complementary determining regions, CDRs, and light chain is also used to have 3 complementary determining regions. So whatever the sequence I got is not matching with the sequence which was sequenced by the virologist. So I need to defend myself. But I have the protein crystals which I used for getting the diffraction pattern. So I went and took this crystal and I gave it to the Maldi person. And they did the Maldi experiment. Finally, they have given me the sequence, whatever is matching with me. And then I reported it to the virologist. And then I also suggested a method for PCR. PCR, there is a method called as a RACE, random amplification of sequences. If you are having a protein of size, whatever it may be, you can design primers, random primers, and if you amplify it, it will give you the full length sequence. And finally, I got my sequence. So only I am telling that this technique is pretty good. Since it worked for me, it will work for everyone. So you can do that experiment, but here in our country, we are not having that many instruments. Only certain institutes are having that Maldi instrument. But it's a pretty good technique. So, and the interpretation is also not that much tough. Once you used to that thing, then it will be easier for you. So this is the outlook about Maldi. So if you have any question, we can discuss about that. hope everyone is doing good. How about your family?
So here we are going to see about like it's.
They are good, sir.
Yes, sir, they are good.
Everyone is doing good. Yeah, because most of your seniors, I came to know that some of them got infected with COVID-19, and their family members also got infected with that. So always take care, take preventive measures, try to get the vaccine. I hope everyone got the vaccine. No one got vaccinated?
So some of us has got and some of us have to wait.
Yeah, for the second
Yes,
dose, I already got Covishield.
So here we are
But
going
my
to see.
second dosage, they are not giving me the time. I don't know, they are telling it's more than 85 days. It's already 1.5 months has passed away. I got vaccinated in the month of April. May has gone. Now June is going on. So finally we will get the thing. Even one dose itself is enough, I think. So a lot more unanswerable questions regarding this COVID also is there. So science is more interesting. Since we are not able to identify each and every day, there are a lot of results are coming out. So far, whatever we are having is not a cure for that COVID. We are just trying to prevent it. Vaccine is the only way. At least we got the vaccine for this. Covishield, the Covaxin, and then Sputnik is already available. And they are working on Moderna to bring it here. Pfizer also ready to give their vaccine. So almost our government wants to vaccinate almost all the population before the end of December. We'll keep our fingers crossed. Okay? I'll try to put the PPT. Let me know whether it is visible or not.
Yes, sir, it is visible.
Yes, sir.
Thank you, thank you. So let's get into this. These are some of the, we look into the proteomics tools. First, we are looking at the molecular biology level, that is cloning and everything, and then separation and display tools. Here also, separation is very important. We are running on the 2D gel electrophoresis. From there only, we are going to cut the band and take it out, and we want to run the protein identification. For protein identification only, we are going to use mass spectrometry. So, what are the main terms we are going to see here is ionization, separation, and then activation, and then mass recognition. Ionization means it's nothing that the protein is injected into the mass spectrometry machine, and it needs to get ionized, or otherwise it needs to get charged. Then only it will travel. And then separation, how it will be separated based on the mass and the charge, it will be separated. Activation, this activation of the protein fragments, that is very important. You need to digest the protein, and then it needs to get ionized, and then only you will spray it. Otherwise, it will not, if it is not digested properly, then it will be a very big fragment, which is not so easy to identify. So, M/Z ratio, based on the ionized protein fragments, only we are going to find out the M/Z ratio. So these are some of the techniques related to MALDI, that is 2D gel plus MALDI, MS, that is mass spec, that is peptide mass fingerprinting. If you are doing it more than 1 or 2 times, then it is called as random MS, they call it as, and then multidimensional LCMS. So where if you want, you can add your isotope labeling and find out. And then from one dimension, whatever the gel you are running it in the lab, you can also take the gel fragment and give it to them. They will find out the sequence, that is mass spec, and they will give it to you. And then we know peptide sequencing is another thing. So if you do not know anything, we can find out the sequence. So this is the simpler look of a mass spectrometry. So where you will introduce the sample into the instrument through the sample inlet, where it is the source, and then it is the analyzer, where the sample will travel, and then finally you are having the detector. Detector is having the electron multiplier, because based on the signal only, you are going to detect it. If the signal is too weak, means then we can't find out that thing. So only we will increase the signal based on the strength. So everything is used to be in a completely vacuum state. So this is the simpler look of the mass spec. This is the basic methodology. How does it work? It creates ions, and then it separates ions, and finally it detects the ions. So okay, after detecting what will happen, it needs to have a library where it will compare the peaks with those library, and finally we are going to find out from the database wherever it matches with the sequence. So this is the thing. So if you look into the principle, so if you look into the periodic table of elements, each element is unique because based on the mass, it is entirely different. So based on the mass, how we are going to find out? This is the basic thing. So if you look into each and every component of a small organic molecule, everything is unique, everything is different. So based on that thing, even if it is like an isomer, it's going to have a different pattern in the spectrum, what we are going to get out of that. So that is another thing. Based on that only we are going to identify the peaks. From the peaks, we can easily identify what will be the protein or a peptide fragment, or what will be the amino acid, and also the small molecule we can easily identify it. So if you have any question, we will discuss. And tomorrow you are going to have the test. I will give you some 40 objective type question. We can have the test tomorrow. That is internal. Okay. Yes or not?
Sir, you have used the PCR, so it will be in silico PCR, right?
What is it?
So you say that you use the PCR for the getting the
You
primer.
are asking about my experiment. Yes, I have done a method called as a RACE, RACE.
So it is in silico PCR.
Of the sequence. Because you can go for the N and C terminal, or otherwise the forward and reverse primer, because we already got the sequence for one chain. It is exactly matching. That is, the light chain is exactly matching with my sequence. And the heavy chain, I was not having the sequence for that. For the heavy chain only, I designed some random primers, and I send it to them, and finally they sequenced it and gave the result to me. Yes.
Sir?
Yes.
Yes, sir. I had a doubt. It's not from it's not from the topic.
Yes.
Yes, sir. I wanted to know tomorrow's test, what exactly will be the syllabus? Could you please repeat? I did not understand it so well.
What will be the topic? It's objective type. I will give you 40 questions. So far, whatever I finished, microarray, pharmacogenomics, and all those things. Based on that, only I will ask question.
So
Okay?
microarray, pharmacogenomics, and I couldn't hear you properly.
Yeah, that's all.
Oh, just these two topics, pharmacogenomics and microarray then.
Yes. But look into the syllabus, it will cover a lot more. But it looks simpler. So go through that thing because it's objective type. Okay? I already sent the
Yes.
PPT. I already sent the PPT to you.
Yes, sir. Yes, sir. Yes, sir.
Okay, go through it. Don't worry about that. Here we are here to help you, and that too in this situation we are not here to threaten you. Okay. So try to write that internals tomorrow.
Yes, sir. Yes, sir.
Okay. Thank you.
Yes, sir.
Thank you. Take
Thank
care.
you, sir.
Thank you.
Take care.
Thank you, sir.
Okay.